Ladies and gentlemen, it is my pleasure to welcome you to today's program, which is the Awareness Seminar of Instal Africa. My name is Rashid Omotayo Adeyemo from the University of Rwanda. It is my pleasure to share the podium with you and to be the moderator for today. And I'll be standing in for our regular moderator, who is Mrs. Aksa Igizadeza. Once again, you are welcome, and particularly a warm welcome to our guest lecturer of today, Professor Amos Abolaji, who has been here on this platform for a while now, waiting for us. It promises to be fascinating, and I'm sure that we are going to benefit a lot from his wealth of knowledge. Ladies and gentlemen, you are welcome once again. Without further ado, let's move on to the agenda straight away. Uh, we are also expecting some of our guest lecture, some of our invited participants, such as Professor, uh, the Principal College of Medicine and Health Sciences, University of Rwanda, Professor Abraham Mikike. We are also expecting to join us shortly the Executive Secretary of National Council for Science and Technology, Dr. Eugene Mutimura, the Director General of Rwanda Biomedical Center, Professor Claude Muvunyi, the Director General of National Industrial Research and Development, Dr. Sekuma, the Director General of Rwanda Food and Drug Agency, Professor Emily Bienvenu. We are also expecting Professor of Human Genetics, College of Medicine and Health Sciences, Professor Leon Mutesa, and also the HOD, Biological Sciences, College of Science and Technology, University of Rwanda, Dr. Mutangana. And lastly, the CEO of Center for Impact, Innovation and Capacity Building for Health Information System and Nutrition, Professor Jinine Congo. I believe that they will all join us shortly. So without further ado, let us move on to guest lecturer. Also acknowledge the presence of Dr. Ms. Inas Ahmed, Dr. Taufik Adeboyega, and Professor Leon Mutesa. You're welcome, sir. I've seen he has joined us. So, and the MD of FIRAT Rwanda Bureau. We have the profile of our guest lecturer. The floor is yours, ma'am.
Dr. Diane is going to give us the profile. Dr. Diane, are you there?
Yes.
Yes, please.
Thank you, the moderator. I'm delighted to introduce the profile of the speaker of today. Professor Amos obtained his Bachelor's of Science, Master's of Science, and PhD degrees in Biochemistry from Obafemi Awolowo University in 1998, from the University of Lagos in 2003, and the University of Calabar in 2010 in Nigeria, respectively. He started his academic career in 2007 in the Department of Biochemistry, College of Science and Technology at the Covenant University. He joined the Department of Biochemistry in the Unity of Molecular Drug Metabolism and Toxicology, Faculty of Basic Medical Sciences, College of Medicine at the University of Ibadan in 2011. He is currently a professor at the University of Ibadan, and he's also a visiting lecturer at the Master's International Health and Tropical Medicine program, where he's at the University of Oxford, where he teaches environmental toxins and the use of Drosophila for detection. Professor Amos Abolaji research mainly focus on the mechanism by which certain environmental toxins predispose to disease such as neurodegenerative disease and cancer by using Drosophila melanogaster as model organism. Accordingly, the focus of his research is to proactively detect health risk associated with these toxins and decode the mechanism of pathogenesis. These would aid in the design of concepts for prevention and possible therapy. Professor Abolaji has published 75 papers in different peer-reviewed Scopus-cited journals. After his postdoctoral training at the Federal University of Santa Maria in Brazil in 2014, Professor Amos set up the Drosophila laboratory at the University of Ibadan from scratch. The laboratory is currently well equipped with state-of-the-art equipment, and he collaborates with scientists from different regions, from the UK, from USA, from Germany, from Spain, and Brazil. In 2018, he received additional training on Drosophila genetics and imaging at the Department of Biochemistry, University of Oxford. He has already trained both staff and students from 18 institutions in Nigeria on the use of Drosophila as a model for biomedical research applications. Since 2014, he has supervised several postgraduate students who have used Drosophila as a model in their projects. He has also co-supervised 4 PhD students and is currently co-supervising 6 PhD students who are using different drugs for the treatment of chemical and genetic-induced Drosophila models of human diseases. Professor Amos Abolaji is a recipient of different grants and funds, such as the International Society of Neurochemistry, CIEN, the Cambridge Africa Arbolada Research Fund, and the CRP International Center for Genetic Engineering and Biotechnology, ICGB. Professor Amos Abolaji has also received several awards, including the 2009 WHO TDR Travel Fellowship to the 5th Pan-African Malaria Conference in Nairobi, Kenya, the TWAS Postdoctoral Fellowship Award at the Department of Biochemistry and Molecular Biology, Federal University of Santa Maria in Brazil, and also the SOT AstraZeneca SOT Endowment Fund Travel Award and the 2018 Africa Oxford Visiting Fellow Award. He is a member of different societies, such as the Society of Toxicology, the Society for Redox Biology and Medicine, the European Teratology Society, the Genetic Society of America, the Neuroscience Society of Nigeria, and also the Africa Society for Drosophila Research. In 2019, he established the Drosophila Research and Training Center, DRTC, as a non-profit organization to facilitate the use of Drosophila as a cost-effective alternative animal model for research and training in sub-Saharan Africa. This research and training center he established has facilitated outreach to undergraduate students from various Nigerian institutions as well as 17 secondary and 2 primary schools in Nigeria to stimulate the interest in biomedical research. The Drosophila Research and Training Center has also trained researchers from different Nigerian institutions on the use of Drosophila as a model organism for the study of human diseases. Professor Amos is happily married with children. We can't wait to hear from Professor the big impact of Drosophila melanogaster in toxicological research. Professor Amos, the floor is yours. Thank you.
Good morning, and thank you very much for the citation. I'm grateful to FIRAT, the University of Rwanda, the management, and all the officials for this opportunity to give this lecture that is titled Tiny Flies, Big Impact, Drosophila melanogaster in Toxicological Research. This is just a brief outline that my lecture will cover. I want to be sure that you are seeing the second slide.
Yes, sir.
Okay, thank you very much. So an overview of toxicology, let me just use the Society of Toxicology definition of what toxicology is all about, which is actually the adverse effect of physical agents, biological agents, toxicants in the environment on living organisms, as well as the ecosystem. This definition also covers the prevention and the amelioration of this adverse effect. It's a multidisciplinary science. It involves different disciplines, different applications to be able to understand the toxicology as a very important field. Toxicology involves a number of things. These are the key areas that toxicology involves. The first one is the fact that toxicology aids in developing mechanism of toxicity of toxicants. In addition to that, toxicology also covers the safety of chemical products and drugs, as well as medicine. As a matter of fact, before any drug will go into the market, the safety evaluation will have to be carried out. Toxicology also involves ensuring the safety of water and food, and also evaluating the risks that are linked with exposure to some of these toxicants, as well as possible therapy. This will guide anyone who is interested in toxicology as an area of specialization. This can guide in how to design different experiments. These are some of the branches of toxicology. It's an evolving area of specialization, and it covers from environmental toxicology to even geographical toxicology, as well as industrial toxicology. The ones that I highlighted are the ones that we currently are carrying out. For example, the environmental toxicology, mechanistic toxicology. There are toxicology that also has to do with the immune system, and even comparative toxicology. Several chemicals and agents that are in the air, in the water, or in the soil, we have several of them. For example, we have more than 80,000 available commercial chemicals. And we have about 2,000 chemicals that has been introduced yearly. And it is important for toxicologists to carry out the toxic impact of these toxicants to be able to understand the adverse effect associated with all of these chemicals. These are air pollutants, sources of toxicant air pollutants. Air pollutant, we have several air pollutants. For example, we have carbon monoxide, being released into the atmosphere on a daily basis. We have hydrocarbons, we have asbestos. These are just few examples of different air pollutants that we have. In addition to the air pollutant, we also have some pollutants that are indoor. And we refer to this also as being hazardous. For example, formaldehyde. This can be found within indoor. Apart from this also, we have several contaminants in the soil as well as in the water. As a result of different industrial activities, some of these waste products actually get into the environment, get into the water and the soil, and eventually this will contaminate the environment. We also have pesticides and nanopesticides as another source of some of these contaminants. We also have micro nanoplastics. For example, the ocean contains about 8.3 million pieces of micro nanoplastics. And then when organisms in the body of water get exposed to these and man consume the fish or any other sea animal, they can actually be exposed to some of these micro nanoplastic. Heavy metals are also there, for example, cadmium, lead, as well as arsenic. These are also very toxic when it gets to the system. Let's take, for example, in Nigeria, there was this report of the lead poisoning incident that happened in the states, in Zamfara State, and as a result of that, several people actually died. In fact, we heard the story about children that are also being used in lead battery recycling factory. And this lead poisoning actually has detrimental effects, even in children, can lead to mental retardment in children. It can also lead to different forms of malformation in developing fetus. I'm sure we have heard about what happened some time ago in Japan with respect to the Minamata Bay, where as a result of mercury that got into the Minamata Bay, and the fish that consumed this mercury taken by man eventually led to poisoning. And as a result of that, several people died as a result of this. So you can see the impact of toxicology in different environments and in different settings. We also have pharmaceuticals, different contamination from waste products from different pharmaceuticals. They can also have adverse effects. We have xenoestrogens that is abundant in plastic and also some cosmetics. These also have different adverse effects ranging from embryonic health as well as reproductive dysfunction. All these can affect the endocrine system and induce several diseases such as infertility, for example. We also have alloalkanes. Alloalkanes are usually used as solvents and also as antiseptics. And these alloalkanes are actually been known to be hepatotoxic and also carcinogenic. We also have atrazines. Atrazines is found in some edible mushrooms, and it has also been shown to have liver toxicity. The smog is just as a result of the adverse health effects in industrial countries due to smoke. When you have smoke, you have a situation whereby there is no clear visibility, it's difficult to actually see, and also if man should get this inhaled, it can actually lead to DNA damage and also mediate different diseases. I mentioned all the ones I've mentioned earlier. It is possible for somebody to be exposed to contaminants from the home, from the work, from different places, agricultural, industrial, and even in the soil. And eventually, if all this gets into the immune system, it can induce different diseases. There are different diseases that patients take to the hospital that are actually as a result of exposure to mixtures of these contaminants. As a matter of fact, we have a specialization in toxicology that is referred to as the toxicology of mixture, whereby they study the toxicity of different mixtures to be able to understand the impact. These toxicants actually have health implications, several health implications. So by way of just briefly explaining some of these health implications, for example, we have what is referred to as teratogenesis. For example, if a pregnant woman is exposed to either drugs or cigarettes or lead, it can pass through the placenta to the developing child and induce malformation in the developing fetus. Apart from this, we also have carcinogenesis, and we also have reproductive toxicity of some of these contaminants. and also nervous system. So you can see the health implication of this toxicant is actually far-reaching. In addition to the health implication, we also have deaths that have been recorded as a result of unintentional poisonings. And you have this in different parts of the world where we have poisoning death as a result of exposure to different contaminants. The fate of these toxic substances as a result of exposure through inhalation or through injection or through the skin, it can get absorbed and then induce toxicity. It can also be stored and bioaccumulate. It can be excreted. It can also be metabolized. So that will now lead us to the different phases of metabolism of some of these toxicants. We have basically, for the purpose of this presentation, I will just focus on these phase 1 and phase 2 reactions. And the phase 1 reaction has to do with the addition of a particular, introduction of a particular polar functional group that will make the toxicant to be more soluble. As a result of that, it could make it to easily get excreted from the system. For example, if an organism is exposed to benzene, for example, an hydroxy group can be added, and then to become phenol, it will be much more polar compared with the benzene. Apart from this, we also have the phase 2, which is actually conjugation, by actually conjugating, for example, GSH with an electrophile to ensure that the compound is actually much more polar than the initial one. Under the phase 1 reaction, we have 3 major reactions in phase 1: oxidation, reduction, and hydrolysis. For students in the undergraduate, you can read more about this phase 1 and phase 2 with respect to the metabolism of toxicants. It's important to understand that toxicologists are interested in different organisms that can be used to study the mechanism of toxicity of different compounds that we have in the environment. And we have had several that have been used in the past that are still being used. For example, C. elegans, which is a worm that is being used in toxicology. We have zebrafish, we have rodents. And also, we also have Drosophila melanogaster, which is the subject of the discussion for this particular presentation. So why is Drosophila melanogaster important in toxicology? The first advantage is the fact that it is easy to actually maintain the fly in the laboratory. In addition to that, it's also affordable. And also, it currently has minimal ethical restriction. For example, to use the fly, you do not need to apply for ethical approval. But to use the rat, for example, in Nigeria and other places, you have to actually apply for ethical approval to be able to use it. In addition to this also, the lifespan of the flies is around 2 to 3 months. And as a result of that, you can study some age-related diseases, such as Alzheimer's disease, for example. And another very important advantage of the fly is the fact that it complies with the European Center for the Validation of Alternative Methods, which is the 3 R's, the replacement, reduction, and refinement. And this makes it actually advantageous to use the fly for study. Another importance of the use of this fly is the fact that the entire genome is well characterized. All the genes present in the fly are already known and characterized. And also, it has about 15,500 genes on 4 chromosomes. If you compare that with that of human, about 22,000 genes in 23 chromosomes. And another vital and striking advantage of the fly is the fact that 75% of human disease-causing genes are conserved in the flies. So the flies contain different disease-causing genes that we have in man. Let me just give an example of what I mean by this. The other time I was talking about the phase 1 and phase 2 metabolism of xenobiotics. And in Drosophila, this is to show that Drosophila also contains some of the genes that humans have. For example, in human, we have the CYP19A1 gene. And this gene in fly is referred to as CYP4AD1. So the name may be slightly different from that of human, but the function is actually conserved in the fly. And that will bring me to state this, that it is very important when you're working with the flies to be able to know the autolog of the gene as we have it in the fly. We also have the FMO5 in man, and in Drosophila we have FMO1 and FMO2. Apart from this also, for the phase 2 enzymes, the phase 2 genes, all these are what we have in human, and we also have this in the fly. For Alzheimer's disease, to also use this as another example, we have all these human genes that are related to Alzheimer's disease, and most of them are also conserved in the flies. This ApoE is not present originally in the fly, but scientists have been able to also express this gene in the fly. One advantage of the fly is the fact that even if the gene is not present and you want to understand the gene, you want to understand the function, you can actually carry out different genetic procedure to introduce the gene into the fly and to express this gene. For Parkinson's disease, these are different genes that have been associated with Parkinson's disease, and all these genes are conserved in Drosophila melanogaster. And it's a very good model that you can actually use to study Parkinson's disease. Another example that I want to give is this particular movement disorder referred to as dystonia. And this dystonia is just a twisting movement as a result of involuntary muscle contraction. And we have different fly models that are being used to study dystonia. These are the different dystonia genes and also the homologue that we have in the fly. So this is important because you can use it to actually know what the autolog of a particular human gene is in the fly, and to also know if it is present in the fly or not. In a nutshell, our research interest in the Drosophila lab at the University of Ibadan is actually to understand the mechanism by which certain environmental contaminants actually predispose to different diseases. And these three diseases we have studied, and we are still studying them in the lab: Parkinson's disease, cancer, and diabetes. And also, when you understand the mechanism of toxicity of some toxicant, it will help to be able to know how to actually treat the disease and how to also carry out preventive measures. We also carry out, we also use genetics to induce some diseases such as Parkinson's disease and cancer, but this presentation will not cover these aspects today. These are the different markers that some of the markers that we actually have expertise in. We have the reactive oxygen and nitrogen species. We have protein carbonides. When an electrophile actually attacks protein, some of the products that you have is protein carbonide. And you can use this to be able to know the toxic impact on proteins. We also have the malon dihydride that you can also use to understand how much of the lipid macromolecule have been broken down. We also have hydrogen peroxide. Apart from that, we also have some of these antioxidants markers that we can study at the level of the gene, and we can also study at the level of the protein or the peptide, as the case may be. For example, for catalase, you can study the gene at the gene level and also at the enzyme activity level. We have GSH. It's important to mention that Drosophila melanogaster can be used to study the impact of different drugs. You can inject this drug to the fly orally, or orally by mixing the contaminants with the diet. And also, you can also carry out this process via injection, by injecting the fly with the drug. It can also be through inhalation. In our laboratory, we actually use this procedure by which we expose the fly to different drugs by mixing with the diet. And then after mixing with the diet, we harvest the flies and use for whatever study we want to use them to carry out. This is an example of a simple device that can be produced to carry out inhalation experiments in the fly. It's a mini review that we have written on the use of the fly in toxicological studies. It is important before carrying out toxicological studies in the fly to understand the aim, the objective, what are the reasons, what are the goal and the vision. Let me give an example. If the researcher is interested in understanding adaptive responses, these are some of the markers that one can look at. If it is to look at, for example, to study apoptosis, these are some of the markers that can be used to. So it's important to understand and to identify the markers of interest before the experiment. And these are different techniques that we can use to study the impact of these contaminants in the flies. Now, let's start with developing mechanism of toxicity of chemicals. Like I mentioned, that the fly can be used to study the mechanism of toxicity of different contaminants. It is important for us to understand this mechanism of toxicity, because if we don't, we will not be able to know how to prevent exposure and to what extent to prevent exposure to these contaminants. This is a study that was carried out by Singh et al. In 2009, and they studied the toxicity, the mechanism of toxicity of benzene, toluene, and xylene, because they actually coexist, and then they carried out studies to understand the mechanism of toxicity of these in single exposure and combined exposure. And as a result of that, the summary of the findings was that these toxicants induce ROS-mediated oxidative stress. And you know, when there is oxidative stress can actually result in different diseases. By way of what we have done in our lab, we studied the mechanism of toxicity of an ovotoxic agent referred to as 4-vinylcyclazine and its metabolite. This was a project that I carried out during my postdoc in Brazil. And 4-vinylcyclazine is actually abundant in different places. For example, occupational exposure. Man can be exposed to it occupationally as a result of, for example, working in companies where they produce insecticides or where they produce tires. Apart from that, it can also be as a result of consumer exposure. For example, people that buy products that are manufactured with the 4-vinylcyclazine, they can actually get exposed to this 4-vinylcyclazine. In rodents, it has been established that the 4-vinylcyclazine is ovotoxic. So we now use the fly to understand the mechanism of toxicity of this 4-vinylcyclazine and its metabolite. And what happens to this in rodents is the fact that it actually destroys the primary follicles in animal model, and this has been established. And we use the 4-vinylcyclazine to study the mechanism of toxicity of, we use Drosophila to study the mechanism of toxicity of this 4-vinylcyclazine. And the 4-vinylcyclazine research showed that it increased accumulation of reactive oxygen and nitrogen species and misregulation of some antioxidant genes such as SOD1 and catalase. And we published this in FIRAT Biology and Medicine. And then we also work on the metabolite as well. The metabolites are the 4-vinylcyclazine monoeposide and the dieposide. And we found out that the enzyme referred to as delta-aminolevulinate dehydratase is also conserved in the fly, and it has 3 thiols at the active sites in close proximity. And when we actually carried out the assay to determine the activity of this enzyme, we found out that the enzyme activity was actually inhibited after exposure to the metabolite 4-vinylcyclazine monoeposide and the dieposide. And as a result of that, we actually used it to now predict the mechanism of toxicity of this contaminant as a result of the fact that it actually attacked the thiol and then as a result of that, it inhibited the activity of the enzyme. Because two mechanisms have been shown with respect to the inhibition of this enzyme. If there is lead in the reaction mixture, lead can displace zinc and inhibit the enzyme. And if there is any other thing that can also attack the thiols, this can also inhibit the enzyme activity. And we published this in Redox Biology. Drosophila is also a very good model that can be used to study the safety of some chemical products and even drugs. And we carried this out some years ago, carried out by Zeniat from the Faculty of Pharmacy, where she exposed flies to posa betas and rusu betas. And this was just a summary of our findings that actually the rusu beta actually reduced glucose level. As a matter of fact, some people that are diabetic also use the rusu beta to manage diabetes. But our finding was, their findings show that it's not advisable for people who have not eaten to actually take the rusu beta because it has the ability to reduce the glucose level. And we did not actually record any adverse effects on the antioxidant status and the behavior of the flies after exposing them to rusu betas. You can also use the fly to study the safety of some food components. You know, some of the things that we use as part of our food components, the fly can be used to understand their safety. We did this for monosodium glutamate in 2017, and we actually found out that exposure to excess amount of monosodium glutamate actually reduced the lifespan of the flies compared with the control that was not treated with the monosodium glutamate. We also additionally carried out study on D-galactose. Excess amount of D-galactose was used, and this actually induced lifespan shortening in the flies. And we use apigenin and to also ameliorate the toxic effect of this D-galactose. We also have another area of specialization with respect to the use of Drosophila in toxicology, where you can use the toxicants that are known to induce certain diseases to also induce that disease in the fly. For example, you want to study the impact of an hepatotoxic agent. You can expose the fly to this hepatotoxic agent and then look at the fat body, which is what is analogous to mammalian liver, and then study this impact. So we have different diseases that we can use the fly to study after exposure to chemicals. These are just some selected toxicants that we have worked with in the laboratory, and we group them in different categories: reproductive toxicants, carcinogens, neurotoxicants. We have worked on all these and other ones that we have worked on in the fly to be able to try to induce a particular disease and to be able to now use what we can use to either ameliorate the toxic effect. For example, we have different carcinogens that can be used to understand and study the process of carcinogenesis. And the work with the fly has actually enabled researchers to facilitate several discoveries. For example, the signaling pathway that coordinates cell proliferation. Drosophila actually provided information and data with respect to this. Mechanism of oncogenes and tumor suppressor cooperation. The fly provided additional information, insight with respect to this. And one advantage of the fly is that when you expose toxicant to these flies and you check the morphology as well as the phenotype on a daily basis, it is possible to be able to see some phenotypic difference when you compare with control. In this PhD project being carried out by Judith, she exposed flies to N-methyl-nitrosourea as well as benzoyl-parine. And the flies that were not exposed to any of these toxicants, you just look at the eyes, the smooth eyes that you can see, but the other flies exposed to these chemicals, you can see the roughness in the eye of the fly. And this is one another advantage of the fly that you can see some phenotypic features that will enable you to be able to phenotypically actually look at what is happening. So each of these can be worked on to now carry out further study to know what are the cancer genes that have been altered as a result of exposure to these carcinogens. This picture was from Dr. Nawe. Dr. Nawe is in London, my personal therapist. That was the company where Dr. Mosura that presented the last lecture work, and they had different flies that genetically modified to have tumor in the eye. This is a particular fly that is treated only with vehicle. And this is another one treated with another drug number 1. I don't know what the drug is. And then this is another fly treated with drug number 234. And then the smoothness actually improved. All of them were like this initially. And that's another advantage of the fly that you can use to screen therapeutic effect of different drugs, for example, cancer drugs. This was a study that we carried out using benzoyl and the metabolites to be able to look at the impact of this carcinogen in the fly. And Oyibo also carried out additional study with respect to carcinogen, sodium arsenite, and then she used this particular plant, Vitellaria paradoxa, to ameliorate the toxic effects of the flies, indicating the advantage of the fly in the study of cancer. Apart from the use of carcinogen, it is also possible to identify different environmental toxicants that can induce some diseases. For example, we have some chemicals that have been known to induce Alzheimer-like. I would like to call it Alzheimer-like disease, because when you're exposed to flies to chemicals, it may not be that disease alone that will be identified. There might be other diseases. And also there will be need to carry out specific biomarker of that particular disease before ascertaining that that disease has been established. So I would like to just refer to it as Alzheimer-like disease. And there are different compounds that have been reported in the literature as evidenced by this review that we published in 2021 with respect to the metabolism and therapeutic relation of biometals that are implicated in Alzheimer's disease. And this author carried out, wrote this paper and identified different areas that we can focus on in case you want to use an environmental contaminant to study Alzheimer's disease. It will be good to understand if that environmental toxicant induces oxidative stress, and to also find out if the mechanism is as a result of abnormal tau or increased expression of the base 1, for example. It's also important to look at the mitochondrial dysfunction. So you study different specific biomarkers with respect to that particular disease is very important. So this we carried out using aluminum chloride that has been established to induce Alzheimer's disease. And we expose flies to aluminum chloride, and we use curcumin to ameliorate the toxic impact. If you look at this title very well, even though aluminum chloride has been established as a chemical that can be used to induce Alzheimer's disease, we did not put Alzheimer's disease in the title because in this particular study, we did not carry out specific biomarkers of Alzheimer's disease. What we just carried out was oxidative stress, which was not actually enough to say that Alzheimer's disease is established. But at the back of our minds, we know that aluminum chloride is a compound that can be used to induce Alzheimer's disease. We also have some reproductive toxicant, bisphenol A and trichloroethylene, that we are carried out in our laboratory. Apart from that, this is trichloroethylene that we carried out in the laboratory. I have mentioned Alzheimer's disease, I have mentioned cancer. Can the fly be used to study Parkinson disease, especially as it has to do with exposure to some environmental contaminants? The answer is yes. I would, however, like to still refer to it as Parkinson-like disease, even though there are some compounds, for example, chemicals that have been known to actually induce Parkinson's disease. And when you carry out this type of study, it would be good to measure the level of dopamine, because this is the hallmark of PD pathology, that in Parkinson's disease patients, we have reduced level of dopamine, but in normal patients, we have normal level of dopamine. So it would be good to actually carry out the level of dopamine to confirm that Parkinson's disease was established. The MPTP is 1-methyl-4-phenite 1,2,3,6 tetrahydropyridine, and is an established neurotoxin that will bring all the features of Parkinson's disease. As a matter of fact, some years ago, people that were accidentally exposed to this MPTP actually came down with Parkinson's disease. And when working with any of these toxicants, it's good to have a very good chemical film yield in order to protect the researcher and the students from being exposed to toxic effects of these chemicals. I have to emphasize the need for high level of safety precautionary measure to prevent exposure to them. And then the mechanism by which this happens is the fact that the MPTP actually attacks the dopaminergic neuron, and then the mitochondria, once they attack, the energy production will be affected, and then it will adversely affect the flies. But when we carry out a 7-day survival, this is the control after 7 days, and you can see all the groups treated with MPTP. You can see significant reduction in the survival rates. And this was a study that we carried out and published in Biochemical and Biophysical Research Communication. Having established the toxicity of MPTP, we now use resveratrol. Resveratrol is found in grapes to see if resveratrol will be able to ameliorate the toxic impact of this MPTP. And we were able to actually found out, reported interesting data. We have also used astaxanthin, which is actually is found in crustacean and other seafood, which is also very effective with respect to ameliorating the impact of MPTP in the flies. We have also worked extensively with rotenone and used different phytochemicals and drugs to actually ameliorate the toxic effect. Here we look at the neurotoxic effect of rotenone that is also added with iron to look at the toxic impact in the fly. And also we use Garcinia cola that is found, colavanol that's actually from Garcinia cola to ameliorate the toxic effect of rotenone that we establish in the fly. This is just an image, video that is just showing us that if we have a control fly that is healthy and another fly that is diseased, for example, exposed to MPTP or any other chemical that can induce Parkinson's disease, the movement is actually altered. I have mentioned cancer and other diseases. Is it possible to use the fly to study hyperglycemia? The answer is yes. As a matter of fact, we have insulin-like peptide in the fly. In man, we have insulin. In the fly, we have what we refer to as insulin-like peptide. We also have glucagon analog secretory cells, which are important in the metabolism of glucose in the fly. This study was published by Ecker in 2017, using high sucrose diet to induce diabetic-like phenotypes, and then to use another plant to ameliorate this toxic effect. We have also carried out this study, and when we carried out this study in collaboration with Dr. Ojo at Wolverhampton University, his lab actually purified margarine AM2 and escalating 2-char. In his laboratory, they have this, and then we collaborated together to expose flies that have hyperglycemia, high level of glucose, and then we found out that these two peptides ameliorated the toxic effect of the excess sucrose exposed to the flies. This has just been published in FIRAT Biology and Medicine, and this was published in 2021, still emphasizing and also showing the power of Drosophila in the study of hyperglycemia. I have mentioned different selected areas of research that we can use the fly to study. For fly researchers, I would like to give this helpful hint that might be of help to you. And one is the fact that it's good to have a research niche. It's good to know your area of specialization, identify it and work on that area and become an expert in that area, instead of having different, spreading your efforts in multiple areas that are not even connected at all. The second advice, helpful hint, is to adhere to institutional policies, understand the policy of the institution, and know how the policy of the institution fits into your goal. You don't just join any organization just because you need a job. You need to understand the policy of that organization. There are some institutions, for example, that put more emphasis on teaching. And we have some that put emphasis on both. And we have some that put emphasis on research. So if your interest is research, you need to know the policy of the different institutions to be able to align with such. I will also give another helpful hint that it's important to avoid intellectual isolation. Don't isolate yourself, interact and collaborate. It's also important, the last hint is that we must be able to replicate ourselves. One of the challenges we have in Africa is the fact that we have some brilliant professors, researchers that have actually carried out innovative researches, but when they retire and when they are gone, you find it difficult to find a replacement for them. It is important to consciously, individually, think about how to raise researchers that will continue with what you are doing and do it even better. And that's part of what we do here from primary school to secondary school to undergraduate and early career researcher. We try to bring down what we do to them in such a way that they will be able to understand. By adventure, one or two of them may pick interest and continue the innovative research that we are carrying out. The second hint I would like to give is, as a result of the limiting research facility in several places in Africa, it's important to improvise, and improvise. I cannot overemphasize this. And I will just give these three as an example of how we have improvised this equipment. For example, these three that you see here are Drosophila incubators. When I checked the price of Drosophila incubators, the smallest one I got was about 5,000 pounds, 10,000 pounds. That's how much they sell Drosophila incubator, original Drosophila incubator, depending on the size. So what did we do? We just look for fairly used chiller. They use this to chill drinks, to make it to be cold. And then an idea just came, and I spoke with somebody, Mr. Eri, that is working in our department, that is it possible to have a device to monitor this and control this temperature, to regulate it from 18 degrees to like 37 degrees, and then to have another device that will help to also regulate the humidity. And after some trials, we were able to get that done. And each of these that can cost several thousands of dollars, we were able to fix these three, for example, with less than $1,000. Another one that we did was when we wanted to buy biosafety cabinets, they are giving us a high amount of price. So we just contacted somebody that actually manufactures aluminum frame, and to just explain what we wanted to do. And this was what he came up with, which was actually less than $30. This is also a chemical film that was locally fabricated. So it's not good to give an excuse, understand the principle of the equipment, and see if you can actually have that locally. I put here the fly-based database. This fly-based database contains whatever you need with respect to Drosophila. Maybe you want to identify a particular gene or a particular protein with respect to Drosophila, or you also want to check if you want to see different fly researchers working on the Drosophila, as well as you want to see conferences on Drosophila and other news that are related to Drosophila. You find it on this database. In conclusion, I need to emphasize the fact that it is possible to have some differences with respect to the metabolism of some chemicals or some drug in the fly and that of man. However, Drosophila remains very useful for discovering toxicological mechanisms that are applicable to mammals. You can have blood-brain barrier differences. However, the molecular mechanism of toxicity may still be relevant in the fly that can be applicable to man. And I will also conclude by saying that when you are doing Drosophila work, think big but start small. Start from where you are, and then think big and proceed gradually. Do not be in a hurry. Be careful to ensure that what you do is reproducible. Positive mindset is also vital. To have positive mindset, to believe that it is possible. It's not just what you do today and you come back a week's time. You have to position mechanism in place to make sure the flies are maintained, they are not contaminated on a daily basis. Our labs are opened every day of the year. During holiday period, people are on roster to ensure that the flies are kept. I want to thank my University of Ibadan, for the opportunity to be a staff at the University of Ibadan. I thank my Department of Biochemistry, and the support from the Department of Biochemistry. Several people that it is difficult to mention what each of them have contributed with respect to the Drosophila research in the institution. I want to thank the current Vice Chancellor, Professor Adebowale, and the past Vice Chancellor, Professor A.I. Olayinka, as well as our Provost, Professor Olayinka Omigbodo, our Dean, Professor Yeronke Odonla, our current HOD, Professor Damoye, Professor Olopade, Professor Olapade Olaupa, and several other people, and Professor Iyo Farombi, my unit director, for their support over the years. I must thank Professor JBT Rocha, who introduced me to fly research from 2013 to 2014. And after leaving his lab, I returned with 2 vials of flies to the University of Ibadan. And that 2 vials of fly has now generated 4 Drosophila laboratories that we are currently using at the University of Ibadan, as well as different laboratories in different institutions that we have worked with. I want to acknowledge and thank Professor Ross Kagan for his mentorship. He has taught us different aspects of the use of the fly in cancer research. I must appreciate his time and investments in our labs. I thank Professor Estavo Cassandra, our collaborator from Harvard University, for her support. Those Africa has also made tremendous investments in Africa with respect to Drosophila research. For example, in 2017, when the University of Ibadan hosted Drosophila workshop, that workshop actually brought about several people who are interested in Drosophila research. And after they left, we had too many people that we can handle at the time. And I want to thank them for their support. Different organizations like Flightbase, GSA, Biochemical Society that have supported us, International Center for Genetic Engineering that supported our project on Parkinson disease from 2019 to 2024. We have completed the project now, and the project was actually ranked one of the 2 best projects for 2023 that was reviewed by IGEP. I learned some things from Professor Ilan's laboratory when he was at University of Oxford from July to September 2018. Professor Ariana Pochisa has been of great support, and I thank her for her trust in giving me the opportunity to teach our MSc students from 2019 to date on environmental toxins and the use of the fly to detect them. Dr. Alex Whitworth has been a very good support to us. We were awarded the Cambridge Africa Brother Research Grant together, and he visited our lab as well, and we learned a lot from him. I thank Professor Andres for some of the lecture that he gave us on Drosophila genetics. The Aid for Labs, we are grateful to them for the donation of consumables to our charity center. I want to also appreciate all the team members, International Advisory Board, and collaborators of the Drosophila Research and Training Center, a non-governmental organization that is interested in carrying out research training to different levels of students at different levels. I'm part of this organization. This organization has actually alleviated, reduced a lot of burden on us so that we can focus on our research and teaching at the university. I thank all the members, current members and past members of our laboratories at the University of Ibadan for the support over the years. I must appreciate you. I thank Professor Adedeji, FIRAT, and the management of the Rwanda University for this opportunity to give this presentation. And I will be happy to take one or two questions that anyone might have. Thank you very much for your attention.
Thank you very much, Professor Amos Olalekan. If we have followed what he has presented to us, he told us about the toxicology, what toxicology involves. He told us about the evolving areas of that specialization with a lot of branches, such that whoever that is interested can key into different, all these different branches he showed to us. He did talk about the sources of toxicants from the air, water, even pharmaceutical products, which has a debilitating medical implications such as endocrine impairment, cancer, smog, genetic distortion, and etc. He did talked about the importance of Drosophila. He said that it is affordable. It is also ethically less scrutinized. It is easier to get ethical approval for the use of flies through many of our researches. It is also, it has 15,000 genes that are on 4 chromosomes. It has 75 resemblance with human genome, which makes it very easy to simulate different human diseases like Alzheimer, Parkinson disease, oncogenesis, and diabetes. He did talk about that the flies are also very easy to study the impact of toxin, which he demonstrated to us, particularly the ovotoxic effect of 4-vinyl cyclohexane. He also talked about the safety assessment of chemicals and herbal products. In Africa, we have this myth that herbal product or product from natural product are not toxic. So he is showing us now, we can see that it is not true that all herbal or natural products are free, I mean the toxicity. So it finally told us about, it gave us some helpful hints, such as the fact that particularly for some of us that are coming to have our own research niche, to avoid intellectual isolation, to improvise. We need to improvise, we need to collaborate, and finally we'll pass the baton to our younger colleagues. He ended by saying we should think big, we'll start small, and be optimistic by being positive minded. Ladies and gentlemen, I believe we must have learned one or two things, and that we might have one or two questions to give to our professor to enlighten us more on what he has presented to us. Thank you very much once again, Professor.
Thank you very much, sir. Thank you very much.
We have a few questions here I can see. First one is, thank you, Professor Amos, for the wonderful presentation. My question is, how can one identify the chemicals that induce a particular disease?
Okay, thank you very much. The first thing to do is to search and check the available chemicals. For example, I mentioned that MPTP and rotenone, they are established and known to induce Parkinson's disease. But MPTP is more specific than rotenone. So some of these have been established the same way. For example, I mentioned that sucrose is known, high level of sucrose is known to induce diabetes. Excess fats is also being used. Cetozozine in rats is used to induce diabetes. So we already have database of some of these chemicals as a result of studies that have been carried out in the past. So you first of all search the literature to find out, if, for example, if you just put it in the internet and you just search for carcinogens, for example, you have a list of them and the study that have been carried out. So we have database that contain all these chemicals and what they have, the disease they have potential to cause. If the chemical is not yet characterized, is not yet known, it can be an area that a toxicologist may work on to understand the mechanism of toxicity and to be able to see if he can identify the disease. But when you are working and you are starting to work, it's good to use an established chemical in the literature that has been known to induce the disease. The advantage of using the chemical is just to be able to mimic that type of human disease and to look for what we can use to ameliorate. Because we cannot expose man to chemical. We cannot use man as an experiment. And toxicologists always look for organisms that we can use to actually model this disease in man. I don't know if the person that asked the question is satisfied with the answer. The person can also send me an email. I can send him a list of some of these chemicals for different diseases.
Thank you, Prof. Thank you, Prof. There's another question. Thank you for the presentation, Prof. Please, how do we confirm diabetes in Drosophila model, just as we measure blood sugar level in rats before treatment commences?
Oh, that's another important and good question. The commonest thing that can be done is to measure the glucose level and compare it with that of the control. And the glucose level, the fly has trehalose, which is 2 units of glucose, trehalose. So instead of the one that we have in man. So you measure the level of the glucose. You make sure that you compare it with that of the control fly. So you can measure the glucose level. You can also measure triglyceride level. You can also carry out gene expression to identify specific genes that are related to diabetes. So it's similar to what you have in man, only that you have to, first of all, homogenize them in appropriate buffer and then centrifuge and use the supernatant to be able to determine the concentration of glucose in the system.
Thank you very much, sir. Another question here. This question is actually speaking my mind. Can Drosophila melanogaster be used to study neglected tropical diseases?
An example?
Let's say Borrelia ulcerans, for example, which is a mycobacterium infection.
That's under infectious disease. The fly can be used to carry out infectious disease. We do not have a chamber and facility to carry out infectious disease in our laboratory, but we have different laboratories within the university that are already up to standard to carry out, to study infectious diseases. So the fly can be used. All you need to do is to expose the fly to this organism and then to now identify the markers that are of interest, especially maybe at the molecular level. Even myself, sometimes they ask me questions that for example, somebody asked me a question sometimes that can Drosophila be used to study epilepsy? I didn't actually, because I've not come across where they use the fly to study epilepsy until I searched the literature and found some publications that the fly has been used to study epilepsy. So you can just put it in PubMed database and search and see if the fly has been used to study that disease. And you can start from there and try to see if you can replicate the findings. So I'm sure it can be used to study some of these diseases. Like I mentioned earlier, that 75% of disease-causing genes in man are conserved also in the flies. So some of these diseases can be studied in the fly. But I am not saying all human diseases can be studied in the flies.
Thank you very much. I think the last question here is from our own Professor Leon Mutesa. He said, is there a Drosophila model used for precision medicine in Parkinson's disease, considering the fact that Drosophila genome contains homologs of several related PD genes?
Thank you very much, sir, for this important question. The Parkinson's disease project that we just carried out is a story that started from the use of chemical to induce Parkinson's disease. After that, we graduated to the use of, I didn't present that aspect, loss of function flies, for example, fly having loss of function in pink 1, packing, as well as fly with overexpression of alpha-synuclein. So we have established this, each of these gene separately. So our next research interest is to collaborate with neurologists and identify Parkinson's disease patients. And then by collecting data, you can identify different genes in a particular patient that predispose them to Parkinson's disease. For example, for cancer, like Dr. Moshura presented, and some of the work we are doing here on cancer, we have identified several cancer genes in different cancer patients, and then we now generated flies having mutation in these genes. So similar thing can be carried out with the Parkinson's disease. Identify all the genes that are already mutated, genes associated with Parkinson's disease, and Drosophila have genetic tools by which all these genes can be mutated in the flies. And then you are looking at the fly, you are looking at the Parkinson's disease patients. And then you can now look for drug that you can use the fly to screen to identify the best drug for that particular patient. So the answer is yes. The fly can be used to carry out precision medicine as it is being done for Parkinson, for cancer. And that is the next that we have in our plan for the future, to be able, after we have worked on this individual gene, to now work on patient-specific PD genes, to now generate flies that we have this. While discussing with one of our collaborators with respect to this, what he mentioned is the fact that if we have up to 10 genes that are mutated, that are related to Parkinson's, and we raise flies having mutation in these 10 genes, that the fly may not survive. But while working with our avatar flies that were working in the flies, we now have understood what to do to ensure that the fly don't die. For example, if the fly will not, there are some of these cancer flies that we're working with that they will not even close. You will not even see the adult. So we start treatment from the embryonic stage, and then we collect the larva and study the larva because they will still be alive at the larva stage, and see if the fly will be able to develop up to adult stage. So it's possible to use the fly to study precision medicine for the study of Parkinson's disease. Thank you so much.
Thank you very much, Professor. Follow-up question to the previous question. I don't know if I should go ahead to ask, but before that, I would implore you, sir, to please probably give us your email address such that whoever that has follow-up questions can always follow up with you. Thank you. So the question here reads, but measuring the glucose level would have killed the Drosophila already. How then can we use that for treatment measurement?
Like we have in rats, you can check the glucose level before the rats will die by cutting the tail and check the blood. But with respect to this fly, what we do is to actually, yes, we'll have killed the fly. But what you can do is different day of treatment. You can have different fly for different days of treatment, and then you check, for example, maybe after day 1, after day 2, different fly. So I don't know of any method by which you can use to check the glucose level while they are alive. I will check. It might be possible, but I've not done that. So what is easy for us to do is to either collect the hemolymph of the fly. The hemolymph, there's a method for collecting the hemolymph of the fly. The hemolymph is just like the blood of the fly. You can collect the hemolymph and then check the glucose level in the hemolymph, and you can also homogenize the flies and check the glucose level. The same way, if you don't want to, if you can sacrifice a rat, for example, you can collect the blood and use to check the glucose level. But with respect to looking at the glucose level while they are alive, I have not investigated that. But if you want to know the level of glucose as the treatment progresses, you can just harvest some flies on day 1, day 2, day 3, day 4, like that, to be able to monitor the progression of the treatment. I'm sure that is what the person wants to be able to identify that. Okay, how do we know that the treatment has been established? So we can do, we are treating the hyperglycemic fly on different days. You can harvest some flies and check the glucose level.
Okay, sir. Thank you very much. Sir, we'll just combine the remaining 2 questions. First is that what is the best attractant to monitor fruit flies? And the second one is, in the case of cancer treatment by plant extracts, how do we ensure this cancer is cured? How to follow the advancements for treatment? Thank you.
Thank you very much. With respect to the attractant, I suppose the person is talking about in case you want to collect flies in the wild. So
Okay.
if you want to collect flies, all the flies we are using in our laboratory are 2 types. One, we collected them from different sources. They are pure strain, not contaminated. Different strain, not contaminated, from different collaborators around the world. We also have another set of flies that we actually set up different lab for. We went into the wild and collected flies in polluted regions. And then we are now looking at the impact of the contaminants in those regions on the flies. The attractant that we used was foods. We used foods like agar foods. We added it to the agar, and then we also use banana. So it will attract them, and then we have a way of trapping them if it is the ones to be collected from the wild. With respect to cancer treatment, if you have established that the fly has a particular tumor, you can treat with your medicinal plants and check the size of the tumor on a daily basis over a period of time. If the tumor reduces, it shows that the plant is working. In addition to that, it's possible also that there are some cancer markers that you can also evaluate, determine in the fly that are associated with, for example, cancer. Take, for example, the P53 gene, the KRAS. You can look at this gene at the level of gene expression, messenger RNA level, compare with the one that are not treated with the flies. You can also look at the oxidative stress levels, some markers specific to cancer. So you can study this in the cancer flies. But if the tumor reduces, it shows that it is working. Like the one I showed earlier in the lab of Dr. Nawe, they use different drug and then focus on the one that will now make the tumor to reduce, and they cannot study it further to understand the mechanism. Thank you very much for all the interesting and vital questions.
Yeah. Thank you very much, Prof. Please, there's one question more, please, sir. This
Okay,
person is asking because it's very peculiar to some of us in Africa universities. The question reads like that: Prof, what advice would you give to people whose institution is focused more on teaching and the staff have only little time for research?
This question is a tough one.
Yeah.
But I will advise the person not to expend too much energy in trying to convince the system or the institution. The person should take responsibility, personal responsibility to do research. As a researcher, as a lecturer, research is compulsory because you discover new things. And if you don't discover, what will you teach your students? It will just be theoretical. And anybody on the field can pick any material and come and read it to students. But when you carry out research, you understand some things, you discover new things. So let me just give a tips about what I did, how I took personal responsibility. When my office, I was expecting an office linked with a laboratory as we had in some university that I've been to. But I was given an office without an adjacent lab. I didn't have a lab. So when I started the Drosophila lab, I converted my office to a lab. I'm not saying you should convert your office to a lab if it's not accepted. This was with the permission of my then HOD. I turned my office into a lab and I started working from there. Because if you have a PhD and you do not have a laboratory, it will be challenging to really do groundbreaking research. General laboratory is for general experiments. Each PhD lecturer or PhD holder is specialized in a particular skill. And that skill may require specific equipment for that specialization. So he cannot use a general lab. He has to have his own lab or our own lab. And as you start gradually from that lab, it will start growing. For example, I told you I started with my office. Instead of complaining, I turned my office into a lab. I supervised MSc students in that lab. After that time, the HOD gave me a bigger space when I told him I needed a space. But he saw what I did with the little that I had and gave me a space. After that time, the space was not enough again. We got additional space. Now we have 4 spaces that we are using for Drosophila research. By taking personal responsibility, the first thing is to have a space. You can plead with the HOD or the dean or the faculty to give you a space. If the space is not available, will they allow you to turn your office if it's within where you can use it as a lab? Or part of an existing lab can be set aside for lab space. So the first advice will be how to get a lab space. The second advice is time management is crucial. I noticed that I have a lot of administrative tasks in the department. So I work up to late hours every day. And also on weekends, I am in the lab. And on Sunday after church service, to maximize my time. Because they pay me for the work I do, I must do the work. For me to be able to now do research, I create additional time by working late on a daily basis and working on weekends. So 2 important things: have a space to carry out your research and also effective time management. And as you do that, I'm sure people of like minds around will see you and they will support you.
Thank you very much, Prof. Before you go, sir, I hope you'll be able to give us some of the vital and comprehensive materials which you have mentioned or during the course of your presentation, sir.
Yes,
And I hope that when next we call you, you'll be magnanimous enough to also share with us from your wealth of experience and your research expertise. Thank you very much for
Thank you very
attending
much.
this seminar.
Thank you
And
for the opportunity.
yes, sir, you're welcome, sir. So the next thing on the agenda is the vote of thanks. Dr. Taufik Adegboyega, please, can we have the vote of thanks now?
Thank you so much. First, on
Dr.
behalf of
Taufik, yes.
yes, I'm on already.
Okay,
Hello.
sir. Yes, we can hear you. Go on.
Okay. Thank you very much. On behalf of the entire team, I would like to thank Prof for the very insightful and motivating presentation. The entire
Hello, your line is breaking, please.
this meeting very memorable. We hope to have you around in
We
our
lost you.
subsequent
Please,
meetings.
can you repeat
Thank you.
yourself? We lost you.
Oh,
Hello?
yes, I
Yes.
said, oh, okay. Once again, on behalf of the entire team, I would like to acknowledge the presence of our professor for a very insightful presentation, as well as the presence of all heads of departments, deans, provosts, and our mentors, most especially Professor Leo Mutesa and the entire participants. We thank you all for making this presentation very memorable, and we hope that we're able to work together in advancing care, particularly with the use of Drosophila melanogaster. Thank you.
Thank you very much. Before we round up, I could see that some might be asking about what is Install Africa. Let me give you a brief synopsis of what Install Africa is all about. Install Africa is the innovation, science, and technology for action and learning in Africa. It's a product of collaborative efforts that started in October 2023. It aims to leverage on the use of Drosophila melanogaster fly model and infrastructure for enhancing fundamental research capacity in Rwanda. It is noteworthy that Rwanda has made significant strides in advancing its scientific capabilities, recognizing research as playing a crucial role in sustainable development and public health improvement. In the view to lay foundation for discovery and development in health, a collaboration was initiated by the Foresight Institute of Research and Translation, Rwanda Bureau, and its technical partners such as Droso Africa, United Kingdom. It also involved the National Council for Science and Technology Rwanda (NCST), the Rwanda Biomedical Center (RBC), National Industrial Research and Development Agency (NILDA), the University of Rwanda, the Rwanda Food and Drug Authority gave birth to this Install Africa. And the goal is to embark on a comprehensive 5-year capacity development plan focusing on training individuals and groups across various levels, including train the trainers programs, the collaborative desire to cultivate the next generation of scientists, and foster research capacity in medical, fundamental, and applied sciences, spanning domains such as healthcare, disease prevention and management, medicinal plants and conservative strategies, agricultural production and food security, drug safety, and developmental biology. The collaboration activities include preliminary tasks such as infrastructure development, grant seeking, mandate assessment, and sensitization effort during the preparatory phase. Subsequent years entail capacity building foundation, research initiatives, symposia, progress assessment, advanced training, and sustainability planning. Collaboration with local and international institutions is anticipated as the consortium evolves. Through this strategic collaboration, this network of institutions aim to drive progress in medical research, foster innovation, and improve public health outcomes, starting with Rwanda and extend to the regions of Africa. Respected colleagues, that is the Install Africa in brief. And thank you all, everybody, for your time. Despite the tight schedule, you find time to be here. Uh, on behalf of our team, this is the end of today's seminar. Thank you very much. Till next when we meet. Bye.
Hi, thank you so much. Thank you, Professor
Thank you.
Abolaji,
Thank you so
for the
much, ma'am.
insightful presentation. We really love that. We appreciate.
Thank you so
Thank you.
much. Thank you for having me, ma'am.
You're welcome, sir. Also thank Professor Leo Mutesa for taking this, for attending this program despite his tight schedule. We appreciate his support.
Bye, everyone.
Bye. Thank you. Our moderator,